them run with their presentation. Please, if you have a question, open up the participant box and you'll see that if you mouse over at the bottom of your screen, you can click on participants. There's a raise hand icon. You can also use the group chat mode as well to put everything, put a question up. And yes, Christine, please mute everybody and stop all videos, and we'll try to conserve as much bandwidth as possible. So Jeff, please.
Okay, thank you so much. Really appreciate all the SMIL team has done for us over these challenging last few weeks to move forward with the annual meeting, bring everybody together, even though we can't be there in person in Ethiopia. And also like just to thank again all my colleagues in Africa for joining today. Okay, and I have the chat window open in a lower screen. So if you're having trouble hearing me or seeing things, and you'd like me to revisit things, let me know. Okay, well, it's a great pleasure to talk to everybody about the progress on Sawagen. And so Sawagen is our new name for phase 2 that highlights the true collaborative nature of this network. So it's improving sorghum adaptation in West Africa with a genomics-enabled breeding network. And here on this first page, I just have listed the co-PIs, but there's many more people who have contributed, and they are acknowledged throughout the presentation and at the end. Okay, so following the template that the ME provided, I'll start with discussion of key activities in 2019. Before I dive into the details of the key activities, I do want to remind everyone of our overall mission, our approach, and the objectives that the key activities tie into. So when we think of the logic chain of this work, the activities feed into the objectives, and that feeds into our mission. And I'll say what I tried to do was focus on where we had previous EAB feedback on particular activities, where we've undertaken activities to specifically address the feedback that we were getting from the EIB. Okay, first of all, the very high level mission. And so this is from the proposal document, and so the high level mission is to coalesce existing R&D capacity, research and development capacity in West Africa into a durable R&D network which delivers varieties that are adapted, adopted, and appreciated. So really breaking that down, we have this input of primarily the existing R&D capacity. You know, we're not really trying to build a lot of brand new capacity on this phase 2. We did a lot of that in phase 1, and we're really trying to take what's already there and just really bring it together in a high performing network. That's the output, is this network and the varieties. So those are outputs. And the outcome, which is how really we want our success or failure to be evaluated, is not just whether we deliver varieties, but whether those varieties can be demonstrably shown to be adapted, adopted, and appreciated. And so we always work back from that outcome. We're not all the way there yet, but I think we can show that we're on track in a lot of respects. So the core deliverable is really this network, and I know sometimes the network, you know, that can seem kind of abstract or vague, but to my mind, it's really not. I mean, it's really the day-to-day of building these relationships, building trust, sharing germplasm, sharing ideas, holding each other accountable to deliver to each other and to deliver to our final stakeholders. The core of the network is this NARS exchange between our 4 NARS breeding programs at ISRA in Senegal, in Niger, in Iran, and then formally adding 2 NARS in the second phase, Burkina, Inara, and Togo, ITRA. And then we really try to make sure that everything that we're doing is just embedded in the context of collaboration with farmer organizations. The proposal lists, I believe, 12 farmer organizations that our NARS members are actively and formally collaborating with. And so the signals of demand come primarily from those farmer organizations and participatory activities that they're involved with. And then the delivery, in a lot of cases, goes through those farmer organizations' seed system. So partnered with farmer organizations, and last and not least, but then supported by these regional R&D platforms. And so this is where CRAS comes in as a regional hub for NARS collaboration. This is where ICRSAT comes in as a regional and global partner, and then KSU. But then a lot of these R&D platforms, I would say, point out, are primarily West African R&D platforms, as you'll see, not driven by the international partners. Okay, where do we want to get to with this R&D network? It's to a network with distinct roles and mutual accountability. So what does that mean? It means that on these development platforms that cover the breeding and seed systems, we free the breeders to really focus on delivering varieties that meet a defined product profile, getting those definitions as rigorous as possible, and then delivering. And so we have a local adaptation breeding platform that is looking at this shorter time horizon, really working with local varieties, not kind of saying whether they're going to be useful regionally or not at this stage, but trying to address some adaptive traits that can be done with more of a backcrossing approach, short time, and then the broad adaptation breeding platform, which is really trying to pick up on some decades of work trying to develop broad adaptation for regional varieties and laying the foundation for long-term genetic gain. Then on the research platform side, so this is where the physiology and the genetics is really focused on delivering knowledge to the breeding programs that directly facilitates their work with the genetic mapping research platform and the physiological mapping research platform. And really what this network is about is the day-to-day responsibility and accountability of delivering across these partnerships, so that in the end, the breeding programs are able to deliver to their stakeholders downstream. We had, one last thing about this. Each one of these platforms represents an objective, and each one of these objectives is led by an early career West African scientist. Objective 1 is to deliver improved versions of locally preferred varieties carrying winning traits using the local adaptation breeding platform. Winning traits are traits that are hypothesized to drive adoption. And that's Jacques Fay, who's coordinating that. Objective 2, to deliver genetic knowledge and tools to breeders that solve their roadblocks using this genetic mapping research platform, which is coordinated by Fanamaina. Objective 3, to deliver physiological knowledge and tools to breeders that solve their roadblocks using the physiological mapping research platform. That's led by Bassin at CIRAS. And then 4, to develop broadly adapted germplasm as a foundation for long-term variety using the broad adaptation breeding platform. And that one is coordinated by Cyril Diaz at ISRA. Objective 5 is the kind of remaining cross-platform capacity that that I'm working to build with the group, or that they're working to build, that kind of cuts across the other platforms. So this is our gender and market responsiveness, overall kind of leadership and management training, and our goal-directed hypothesis-driven scientific method. That really is the glue that holds all these objectives together and brings them into a durable network. This goal-directed hypothesis-driven science, we've been working really hard on that. This is a screenshot from training material for Go High. If you're interested in these, learning more about the details, it's available at gohigh.org. I'm just going to block that out, because I would really be happy to do more training with anybody who wants to do that. I'll block out these details, but really it's a rigorous logical chain for goal setting above and connecting that to hypothesis testing below. And where does this thing come from? Well, a lot of it comes from me trying to reverse engineer and formalizing the goal setting that I learned from phase 1. So a lot of that was critical feedback from the EAB. You know, when I think of all this feedback I got from the EAB in the first couple years, in terms of Go High, you know, I think like a lot of, you know, new faculty members, I was very, very focused on my, you know, the detailed objectives of all the stuff that I was an expert in from, you know, from my previous work on GWAS and genomics. And a lot of the questions from the EAB were like, how is all this detailed stuff going to connect back to the impact you want to have? And so really what I want to do now, what I'm trying to get everybody on the team to do, is don't work backwards from the detailed objectives, work forward from the vision that you have, and step by step how you're going to accomplish that. I learned a lot from breeders with a record of adoption of their varieties, number of those on the SMIL team, and tried to reverse engineer how they think. And then really tried to formalize what Tim was allowing, you know, me to do, what he was doing with me, adaptively managing. You know, I realized he was allowing me to change objectives because I was describing to him hypotheses and how we learned something new about hypotheses. And so that gets to the second part of it, which is connecting goal setting to the scientific method. So that means a priori, before the fact, defining competing hypotheses, predictions, and doing this over and over again. And I believe what Platt said, that great science is not due to genius, it's due to good habits. Anyone can do it. And so what we're trying to do is good habits over and over again, using the scientific method and connecting it to our goal setting. Okay, so one of the most important hypotheses we have is that the varieties that we're developing are responsive to needs of both women and men farmers, responsive to the needs of youth, and responsive to other needs in the market. And so one of the key bits of EAB feedback was gender sensitive targeting should be integrated into project activities. We completely agree with that. And so one of the major accomplishments this year is that every NARS breeder, along with the social scientist from their own NARS, has had gender responsive training from the GREAT program. And this was actually a part of what I think is the first regional training that they did in French in Burkina. So this was like a really major regional capacity building that was instigated, partly by the Sawagen team. So I want to thank the folks in Burkina who arranged that with the GREAT team, and of course the GREAT team led by Eva Velstein, our mentor, and Holly Tufan. Maybe this might be a good time to bring in. Does anybody who attended the GREAT training want to highlight a key point about that for the EAB, what they learned? If the connection is good enough. Let's see. I see Cyril is on. Any of the other breeders are on. Cyril, do you want to comment on that?
Do you hear me?
Yes.
Yeah.
Okay. I comment this training. I participate with one socio-scientist, Muhammadu Rasul C, to this training. It was a very, very important training where we learn how to integrate gender in our breeding programs. I know that previously in my program, gender was not really integrated based on the understanding I have during this training at Burkina Faso. During this training, we have a chance to have Eva. She have a very huge experience on gender responsive breeding in Mali, and he shared with us many case studies during this training, and I hope we learn a lot.
Thank you, Cyril. And the EAB might be familiar with this program. This is a really rigorous program. It starts with a week of training, but then continues with follow-up activities over a year, where the breeder and the social scientist are really integrating fully what they learned into their program, and doing that with experiments. Okay, thanks, Cyril. Another thing that we've expanded a lot is multi-environment evaluation of the material from the mini-NAM from one. One of the bits of EAB feedback is more effort needs to be made to get phenotyping right, and we'll talk a lot today actually about the core breeding activities, but really, it does come down to crossing, selection, phenotyping, right? And so we agree, and we're working hard to really test the representativeness and the quality of our phenotyping in a lot of different ways and on multiple dimensions. So a few key activities related to that. Right now, it's expanding on-station breeder evaluation across of multi-environment trials across. Sorry, that should be West Africa, WA, and the participatory and on-farm evaluations are planned for the future, yeah, so I think Ardali is not, I don't think we have somebody from Niger on the call right now. So maybe I'll just pause here. I don't know if we have Nofu. Cyril, do you want to comment at all about the expansion of multi-environment trials in the network?
Okay, let me. I took advantage of the GREAT training to visit the program of NUFU at Kambwanse. This year, I share the inbred lines I get during the phase one. During the phase one, to recall, I identify some QTL for grain mold, and with using a F3 population during the phase two, based on the families that have QTL, I selected 25 inbred lines having QTL for grain mold, and I'd share seeds of these 25 inbred line to Burkina, Niger, and Togo. During the grid training, I visited the trial, the Mininam promising inbred line for grain mold resistance. This picture shows this, there's lines in Burkina Faso. And globally, we found that all the varieties of all the inbred lines in Burkina Faso have a good behavior for grain mold resistance. I hope it is a very important result for that.
Thanks, Cyril. Yeah, so that was the issue of grain mold, quality of the grain mold phenotyping, and the panicle structure. That's one that we've talked about a lot. We got a lot of feedback from EAB on that. So Cyril is working to open up the panicles back, learn from the experience of the smallholders, and also we're really more broadly evaluating grain mold resistance on the current material. We're taking both strategies moving forward. Okay, and then following on this theme of the multi-environment evaluations, one of the bits of EAB feedback was that the payoffs will be growth and a lot of work that we're doing to building the collaboration, and that's definitely true. And we also do want to be held accountable to deliver soon. So we appreciate the benefit of the doubt, but we also want to prove to ourselves that we can deliver. And so we are, as Cyril described, we're moving forward on the material from the phase one mini-NAM from Senegal. Niger was just a little bit behind Senegal because they started a little bit after. So, but there's material from the Nigerian mini-NAM that has already been selected based on farmer and seed company evaluation, and I'm going to just present this because I don't think anybody's on from Niger. Yeah, it looks like they haven't connected. So they are now advancing that material this past year to F7, so to get those pure lines for multi-environment evaluation, and all the work that Fana's been doing, and I'll mention a little bit more, or Fana will if she's on the call. To develop these Striga resistance markers based on Gabisa Ajeta's work. That now has led to the capacity for INRAN to use outsourced CAS markers to do marker-assisted selection on this material. It's cross of a local variety MDK, Masse de Cunha, with SRN39, Gabisa Striga resistance variety. And so Fana is going to be doing marker-assisted selection along with the phenotypic selection in the Striga phenotyping facility that Aissata and Ardali run in Coney. Okay, on this issue of getting the phenotyping right, one of the big ones that we're focused on is validation of managed water stress and multi-environment trials to capture the target population of environments. And so in these activities on Objective 3.1, we're directly testing the hypothesis on the utility of phenotyping approaches with a few different, from a few different directions. One is head-to-head comparison of plant performance in field versus semi-controlled systems. And so I won't go through the whole hypothesis testing logic here in the interest of time, but we have gone through with GoHi and developed a priori hypotheses and predictions about plant performance in the field versus plant performance in the lysimeter that will allow us to reject the hypothesis that the lysimeter phenotyping is useful directly for phenotyping, if that's the case. And so we're not making assumptions about the utility of any of the phenotyping that we're doing. We are systematically testing each of the phenotyping approaches. And I'll just point out that this would be necessary, is going to be necessary to do even when we're on farm, because no one farm or no set of 20 farms is perfectly representative of the TPE. Always we have to test hypotheses on their representativeness. So this was done in 2019. Falalu Hamidou and Abdu Haru and his group have just completed this experiment in FANA, and them are analyzing the data and then testing the hypotheses. Okay, on the same lines, you know, the other thing we're doing to test hypotheses on the utility of phenotyping approaches is to test hypotheses to look at predictions of that hypothesis on the utility of how well our experimental systems basically map to the TPE using ecophysiological modeling. And so that's work that Bas and Diaretu have been working on with the help of Vincent Vadez. And here's some of the recent trials to calibrate those models. Let's see, Bas, I believe, is on the line. Bas, do you want to comment on this? If your connection is good enough. Maybe Bas can jump in, or Bas, if the connection is not good enough, go ahead and put comments in the chat window that we can highlight. And I see actually he's added a another point that there's a modeler based at Bombay who was on the left in this photo, is also involved in this work.
Do you hear me?
Yeah, it sounds like the connection is not great, but let's give it a try. And if it's not good enough, you can just put your comment in the chat.
Okay. Then what we want to do at this point is to test if the work that we are doing at the off season, in cold off season, is representative of the drought that we are facing in the rainy season. For that, we envision to parameterize some varieties, a set of diversified variety from the WhatsApp, and to parameterize this variety in the Samara model, to run the model in specific rainy season condition when we have drought, and compare this result, particularly the biomass and the yield, to what we obtain in the off season, and see if we have the same comparison of the variety. That's what we want to do, to test if the work that we are doing in a physiological aspect in the off season is really representative of what we are obtaining in the rainy season.
Thanks, Bas. And so, as Bas described there, we're not assuming that the managed stress in the off season is representative. We're taking that as a hypothesis, and we might reject that hypothesis, and then we will adjust our activities accordingly. Okay, so So under that local adaptation breeding platform, right, this is where we're doing a marker selection to be able to rapidly develop some new candidate varieties that will then move into phenotypic testing. There's been a lot of work on genotyping, and so this is an example of the team at ISRA collecting leaf punches out of the breeding program. And in this case, you know, so I'm going to come back to this later. I think Danielle and Cyril on the call, but I'll ask you to come in when I talk about the changes that we've made. But here they've made crosses between these local varieties with B35 as a potential source of useful traits, in this case, stay green, drought tolerance, and that's being sent to Intertech for CASP genotyping. So that's a collaboration between Cyril and Jacques Fay. And we'll come back to the CRS Biotechnology Lab in a little bit, and I'll ask for comment from Cyril and Danielle then. Okay. Okay, so that is the highlight of the main activities, especially on the West Africa side. Moving to obstacles. So one of the things that we were doing in the second phase is trying to integrate additional CRS research activities in. So a couple of people reached out to us that were interested in really integrating what they were doing at CRS into the Sawajan team, and we were very happy to do that on the molecular biology side, Amadou Diallo, and on the crop modeling side, Diaratu Sambake. We haven't done quite as much of that as we expected. Amadou got a new, well, he's working for the ministry as a technical advisor. He's been, I guess, seconded to work for the ministry for an indeterminate amount of time. So, so his work is on hiatus. Diaratu was on leave for part of the year. So she's been working with Bas, I think, closely, but we haven't had as much interactions with the rest of the team as I anticipated, but that's pretty much, that's all fine. We'll just pick up these activities as we go. This is just long-term kind of building the durability of the network, so that's not really a problem. An area where we're really going to focus in the coming years is just getting these breeding program fundamentals solid across the whole network. Everybody's well trained, but everybody's starting out, and so there's a lot to learn when you're running your own new program. I've definitely learned that over the last few years, and so we really just realizing we need to make sure that everybody across the board has their breeding program fundamentals where they want them to be, and so that's just things like crossing success rate, seed lot purity, and identity. And so I want to talk about the expansion of quality control genotyping. And at this point, let's see if Danielle was able to get on. Yes. Danielle, can you comment on that part of the program?
Good morning, everybody. So we at the Service Lab, we committed ourselves to try to perform the quality control of all crossing that leaders are doing in their program. We started with Togo crossings, and we tested all F1, the putative F1 produced by Jakarta along with the parents, and so the results we have. So Jack got some good F1s hybrid, but the parents was not the true parents. So that pointed out the issue of security and probably the way they are controlling how they are bringing the pollen into the parents. So that was first the program. So we tested also the work done by Jata, F1 he produced. So Cyril has some much good result than Jack, but again, we found that there is some issue of seed loss purity, and sometimes the parents they think that they are crossing are not really the one they will. We discuss a lot with Geoff and also Jacques Martin. We also tried to compare what we were doing with the SSR markers with the Intertech SNP genotyping platform, and the result was quite the same. So we validate that there's some seed slot the purity issue in the breeding programs, and that we need to generalize the quality control of the genotyping at each step of the breeding, to be sure that the hybrids that are being produced are true hybrids, and that they are coming from the parents that we want to cross. So that's the work we were doing. Now we received from Niger by Ardali Husseini, but because of the COVID outbreak, we don't have any more access to Sarah's lab. So we are waiting that the situation evolve in the good way to start to control these crosses.
Okay, thank you, Danielle. So, you know, then like the thing that I really have loved about this interaction with Danielle's genotyping lab is it's really driving natural, organic NARS collaboration on these breeding program fundamentals. It's not like me going and saying like, oh, you know, do your crosses this way, or, you know, this is how you should be managing your seed. They're sharing germplasm. They want to know what it is. They want to make sure that they're managing it as well as possible. And I don't have a slide on this, but Cyril has been working really hard on implementation of breeding management system, and the others have been implementing it. And so all these things work together to get the breeding program fundamentals reinforced. And when there is weak points, you know, we address it as a team. You know, it really helps bring these issues to the fore so they can be addressed. Okay, let's see. Okay, so moving on to achievements and lessons learned. Big achievements in progress as we speak is these KSU PhDs. Fauna just defended last week. Jacques next week. I don't think Jacques on the call because today's his deadline for submitting his thesis to his committee. I'll just highlight a few key findings from them on the gene pools for West Africa sorghum breeding germplasm, on drought resilience and striga resilience. These are all collaborations with the West African-based team, but these are analyses done by Jacques and Fauna. So one thing I want to mention, this thing of building goal-directed, hypothesis-driven culture. It's not a one-time training thing. It is just week after week, day after day, getting into good habits, holding each other accountable for doing rigorous goal setting and connecting it to rigorous hypothesis testing. And so one of the ways we do that is with these bi-weekly Zoom meetings. And so in this past year, we've had 16 meetings and we averaged 6.3 attendees. I put the 0.3 so you know that we actually took attendance, and I calculated that. So we have a breeding meeting and a research meeting each month. And the breeding meeting, the focus is every time it's where are we at on breeders' delivery of varieties to seed system? What do we need to be doing to help the breeders to make that happen? So we go down into the details, but we never forget why we're there talking. The research meeting, the focus is on the researchers' delivery of knowledge to the breeders that addresses their roadblocks. And each of these meetings is focused on either breeding or research, but that doesn't mean that only the breeders or researchers attend. The breeders and researchers attend both meetings, and they build linkages that way and understand each other's programs and how to deliver from one to another. Here's a screenshot. This is kind of meta. Here's a screenshot on Zoom of a Zoom meeting. Due to COVID contingencies, due to COVID contingencies, Fauna just defended on Zoom. Maybe the first successful defense on Zoom at the university. I think we were the leaders there. And Jacques is up next in in a couple weeks. And I just see Peter has a question. Would you like to address that now, Peter, or do you want to do it at the end?
Oh, we can do it at the end if you want.
Either way, I just have a couple more slides, but why don't I keep going? Then we'll be done with the slides in about 3 minutes. Okay, this is some of Jacques' work, taking this genotyping that we were doing and really getting it down to take-home messages for the breeders. So one thing that the breeders really care about is what are their gene pools across the different programs. And so here we have color-coded a plot of basically which gene pool you are is which color. And you can see now the breeders have a accession by accession map of, these are the caudatum genotypes in red. Which are the similar caudatum genotypes in the different countries. And so that's a part of Jacques' paper describing the West African Sorghum Association panel from phase 1. It also shows that there's some unique gene pools that are really only prevalent in one of the countries. And so the management of the germplasm, the management of the, who do you want to cross with if you're trying to establish elite gene pools for your country or for your region. Now there's kind of a map of which accessions belong in which gene pool. These are some results from Jacques Fay based on the phenotyping data from Bas and Jacques Akata in phase 1. Jacques Fay has now clearly demonstrated that maturity 6 is the major gene controlling photoperiodic flowering in West African sorghum. It had been cloned in Texan material, but we had no idea what the genes that were controlling photoperiodic flowering across West Africa were, and he very precisely mapped that. This is the MOS6 gene, and this is the peak association here. And it may explain up to weeks of the variation in flowering time that are caused by photoperiodism. This is probably the most important adaptive trait in West Africa. I think most people would agree with that. The Malians have worked a lot on that. ICRISAT has worked a lot on it. And it's actually really slow and time consuming for breeders to select phenotypically because you need pure lines. You can't do early generation selection. You need multiple planting dates or multiple locations. So the breeder demand for a marker is there. And so this is something that Cyril and Jacques have kind of worked out among themselves, and they just told me about it. And I was like, so excited, because this is exactly what we want. We want geneticists to respond to the breeder's demand. Fauna, using the data from her collaborative experiments with Falalu at ICRISAT, has identified what appears to be a novel drought resilience locus, along with potentially a novel drought resilience mechanism that's based on 2 years of lysimeter phenotyping of 200 WASOP accessions under managed stress. You see this peak in the genome-wide association study is particularly pronounced under water stress. It's also there to some extent under well-watered. What she noticed was that this SNP is associated with reduced vegetative biomass under water stress and increased grain weight under water stress. This exact same SNP was identified. And so it's relatively common in the WASOP. So it's already there in West Africa. But what the new hypothesis is that this SNP tags a variant that controls our assimilate partitioning to the grain under drought stress. And so this is the type of kind of in West Africa discovery that we were hoping to be able to do. And this is a hypothesis, nothing more, nothing less, but it's a very testable hypothesis. We can move this allele easily from one West African line to another. And it's already there in West Africa, but it's not present in all the germplasm, right? In 86% of the West African germplasm does not have this. So this is an opportunity to move kind of smallholder technology from West Africa, loci that smallholders selected in the past, to other West African smallholders via the NARS collaboration. And then the last science update is on just Fauna's work on trait validation for Striga resistance. This is an example of this really rigorous hypothesis testing that we're doing now. Fauna and Jacques have been working really hard on that, and we're working with the rest of the folks in West Africa on that as well. So she sets up these competing hypotheses right from the beginning. LGS1, the low germination stimulant gene that Gabiza discovered, does confer resistance to West African Striga and West African varieties, or it doesn't, right? And it's really possible. Hypothesis B is possible for a variety of reasons, like ecotypic differences in Striga, or because of gene to gene interactions, that maybe this allele doesn't work in different genetic backgrounds. So she sets up her experimental design, and then she a priori sets up the competing predictions under each of the hypotheses. I won't go through those, but now she's gone up to Penn State. Might be the only Striga quarantine facility other than Gaviza's, I don't know. But Jesse Lasky's lab, she spent a week with them working on testing this hypothesis. She's supposed to go back basically now. That's been canceled or postponed because of COVID. But we are not taking anything for granted. We are very skeptical about our own work, and we're using this scientific method to rigorously test everything we can. Okay, future planning. The breeder focus really is on those fundamentals. So getting those product profiles really formalized into hypotheses on stakeholder demand. We've done a lot of work on product profiles, and we'll continue to do that. Formalize these hypotheses on phenotyping, whether the phenotyping we're using, no matter what it is whether it's representative of the target population of environments on farm. And then what Danielle talked about on crossing success and germplasm management. And then just keeping focused. Gito mentioned minimal viable products. It's very easy to take on too much, and we're trying to keep an eye on that. On the researcher side, just really complete that delivery of the units of knowledge to the breeders, making sure that those phase 1 publications get completed, and making sure that they have take-home messages for the breeders. You know, it's not good enough just to make the breeders wade through all the data. we want to speak directly to the breeders in our written communication in those publications. We've done a lot on the Go High training, and I think we need to do more on addressing those kind of weakest links in the logic chain. A lot of the Go High process, people are already familiar with parts of it, but we need to figure out where people are less comfortable and make sure that that's where we don't skip over the logic. We really focus our efforts on those weakest links and really using it day to day to get good habits. And something that's can be painful, but I think we need to keep in mind is really dropping research that isn't a breeder priority, or whether our hypothesis testing shows us that it's not going to impact the breeders. COVID contingency planning, last one, is we're looking at delaying Fauna and Jacques' return to West Africa. They're getting done, they will be done, but we can just delay the final thesis submission to the grad school, and that would allow them to continue on a GRA through the summer. Jacques and Fauna and I were going to go to WACI to do a course on goal-directed hypothesis-driven science using R programming this summer. That's not going to happen. And then Fauna's follow-up work at Penn State in the quarantine facility, postponed. Hopefully could happen later in the year. And then obviously everything's up in the air for field season and participatory work, so we will stay posted. Thanks to all the team. Hopefully I've noted everybody here and our advisors and our mentors on the project as well, and we'll take any more questions.
Great, thank you very much, Jeff and team, for your comprehensive presentation. We'll open up the floor to questions. One's already been chatted a little bit about on the while you were talking. Peter Matlin had some questions on, do you have any hypotheses on the main factors contributing to seed lot purity problems that you identified by SSR genotyping? Were there differences across the breeding programs? And what were some of the most important contributing factors and steps needed to resolve? Daniel's already talked a little bit about this in his response, in his type response, but for everybody's information, maybe we want to just talk about that because I find that interesting as well. Daniel, do you want to just chime in with and explain a little bit on what you wrote to Peter?
Yeah, for the question asked by Peter, it's something very frequent in the breeding programs, at least for West Africa, and we tested it for many different crops, including cowpea, groundnut, millet, and sorghum, of course. And what we have seen is that when some lands share similar features, for example, seed color or some same cycle, or for same crop architecture, etc. People are mixing it very oftenly, and sometimes they just don't have the means to distinguish one land from another. So it's something very frequent. And for example, in my breeding program, all the parents I'm using for crossing, I'm trying to ensure myself that they are coming from the same seed lots that have been produced from one unique plant, etc. And it's not really the same practice in other breeding programs. Sometimes they have some line, so they sow many plants in one line, and they do their crosses with different parents, etc. And the more seed you use in your crossing, the more risk you have of mixture. So it's something that is very frequent, and we observe it for different crops in Senegal and in West Africa. And the difference between the breeding programs, it's really relying on breeding program maturity. For example, Togo is very a young breeding program who Jack is trying to push this breeding program forward, but it's something that have been started, I think, a few years ago. Probably it's 3 or 4 years. But the one in Senegal have a kind of better knowledge of their materials. That's the reason why we have some good difference between breeding. And in Senegal also, most of the lines have been also fingerprinted. The lines used by Cyril, so we have some reference, and we can check sometimes the lines for purity, even if we are not doing this in a systematical way. So right now, what we decided to do is to systematically check all the seed lots for purity and develop some seed lots especially for crossing, and follow them in the right way. So that's the one of the comment I wanted to put on it.
Thanks, Daniel. I'll just add, this is not anything unique to West Africa, and it's not, in any way, saying that the breeders are not qualified or doing a good job. Steve Kresovich, a mentor of mine, told me that when he was overseeing this for GRIN in the early days of genotyping, his rule of thumb was that 20% of the pedigrees weren't going to match. And so it doesn't mean that anything went wrong in these breeding programs. It might have been something that happened 20 years ago. It's the pedigree information is only as strong as the weakest link. But they are working hard to improve all those aspects.
Great, thank you. So other questions for the team, you can raise your hand or use your chat box. Bettina's got a series of questions. Jeff?
Yeah, so
one question was,
oh, go ahead.
or you can read it.
No, go ahead. Why don't you do it verbally?
Yes, one question. We are in contact with the SMIL-funded project on sorghum disease resistance in Niger and Senegal by Clint McGill. Like they are also working on grain mold.
Yeah. Okay. So the quick answer is yes. So I had a lot of conversations with Clint at the last SMIL meeting in Niger, and actually even before that, he had been reaching out to me for collaboration on the genomic side. I think he mentioned that at a couple days ago. So I'm interested in collaborating with him. I think actually, to be honest, the biggest challenge for me now is that they are not using this kind of go high type approach where they're like identifying the breeder roadblocks. And so it can be a bit difficult to connect all the work that they're doing to the work we're doing with the breeding program. I would be interested in working more with them on that, and I guess probably Cyril is the best person to address the question further, since he is on both teams, and he can comment whether that go high issue is part of it or not. Cyril, do you want to say anything about that?
In Senegal, I'm working with Herman Sar, who is the phytopathologist. And during the prom visit in Senegal, I did it with prom. All what he mentioned last days, I did it with prom. So the survey he did on Senegal, it is with me. So we are collaborating on grain mold specifically. Currently, we don't know exactly the different species involved in grain mold. And this work, we are doing it with MAMP and SAR, who is a phytopathologist based at Cienara Bombay. So we are there connecting the two projects.
So
Okay, that's great.
I'll just make a comment about that. You know, from my perspective, the pathologist working with the breeders is maybe necessary but not sufficient to be like useful for the breeders, in the sense of like if there isn't a like roadblock for the breeders that the research is helping with, the breeder could spend a lot of time helping the researchers, but then it doesn't necessarily feed back to help the breeders achieve their, you know, their varietal development goals. And so that's the thing that, you know, I would be really interested in coordinating with them on. You know, it's not, you know, obviously it's not my project. I don't have, you know, a mandate to do that myself, but I'd be interested to do that if they're interested. I think Boss is ready to answer the second question. Bettina, if you want to ask that.
Yes, I think the answer is also already in the chat. The effect of day length differences in the off-season versus rainy season, that could also affect the response to drought stress, especially in the photoperiod sensitive varieties. And Basiru said, yes, cycle length is shortened and there may be a bias.
So, okay, so this is the type of thing when I only showed this one work through example with striga resistance. This is the type of thing that we are spending a lot of time on now, where previously we and a lot of other people kind of hold on to one hypothesis really strongly. So like we might want to hold on to the hypothesis that the managed stress phenotyping is not biased, right? Because we've invested a lot of effort in that, right? And so the EAB member says, oh, but maybe you need to consider this other hypothesis that it's biased. So what we're trying to do is just do that beforehand and not get overly invested in one hypothesis or another, and figure out how to eliminate one or other hypothesis as quickly and efficiently as possible. So we are conducting tests to really try to define what biases it might cause. You know, and we are also considering, and based on those tests, you know, if the managed stress is not, if those tests are showing the managed stress is not relevant, we will reduce the amount of managed stress, you know, that we do, or even eliminate it on the project.
Okay.
Yeah, I think that's a really good approach, the hypothesis-driven research. I really appreciate it. Thank you.
I'd like to just go back to your first question, Bettina, on the linkages between the two products. And Gito, is there anything that you can see from your perspective as the leader of CIRAS that you could, any steps you could take to encourage tighter linkages? I can certainly see at the management entity standpoint, we want to try to facilitate a space between the two projects to interact, but what about operationally at CIRAS? I see you going. Now you're muted. Try unmuting, please.
I just did. Let's see, let me try again.
You want me to speak or what?
Yes, yes. Do you have any, I guess the point that Bettina
Okay, thank you.
was on those linkages.
Yeah, was it about the question of Bettina or what?
Yes.
Because
That's Bas, not Gito. You were asking Gito, right?
Right, that's right. Sorry, who's speaking? That was that boss? Yeah, Guido.
Yeah, Bas, Tim was asking of Gito about the overall coordination at the CRAS level. Oh, so Gito's is asking asking you, Tim, to type in the question and he'll respond over chat.
Okay, both team members are involved in the projects. It's certainly a start.
Yeah, so I will, I'll just say Gito and I have been working a lot together on Go High implementation. And so, yeah, so one of the things that Gito and I have been discussing is how basically people working together is like necessary but not sufficient for delivery. And so one thing that happens is people will work together. Like, let me take this as an example. I'll do a counterfactual. So I hear Tim Dalton says, I want to see SMIL projects working together. Okay. So then I go, okay, I can have somebody in my team do the GWAS for Clint McGill. You know, they're not specialists in GWAS. We'll just do it. And so then everybody's happy because we're working together. Everybody can report how great we're working together. But that's down at this bottom of objectives. It's not clear whether logically doing a GWAS addresses any roadblocks for Serial's program. It might, it might not. And so what Gito and I have been working on is kind of setting up kind of how long-term we work together to make sure that whatever research activities go on, they're clearly tied to the roadblocks for the breeders.
Good, great. Questions from the audience? Either raise your hand or type them in, depending upon your bandwidth.
May I come in? I have a question.
Yes, please go ahead, Berhani.
Well, this question I have is to Jeff. I think, as we all know, this is one of the biggest projects of the SMIL in phase 2, involving 4 different countries and so many different scientists from the US and from the region. I think it's a very ambitious project, and I hope there will be a footprint of this project, you know, when SMIL terminates 3 years, 4 years down the road. The question I have to Jeff is that, you know, when SMIL really terminates, what strategies do you envision that the overall the Sawagan project or the concept, the approach will continue after the project ends? And I think we need to think of ways that the footprint of this project will continue after the project ends. Can you comment on this, Jeff, how you envision this?
Yeah, thank you so much. Yeah, absolutely. And so I'm very, very happy to report that we have now majorly funded an opportunity to take the lessons learned from Sauergen and from SMIL more broadly, and to scale them up, both in terms of geographies and in terms of crops. And that is for this new Feed the Future Innovation Lab for Crop Improvement that just started. So this is a $25 million innovation lab. I'm the lead for trait discovery, one of the 5 areas of inquiry, or now they're calling them objectives. So I have a full-time program manager now who just started a couple weeks ago, Tarek Aluri. He's on the call right now, and his basically his full-time job for the next, say, like year realistically is really to learn about what has been done on Sauergen and SMIL and other kind of projects like them, and distill that into tools and best practices on this innovation lab for crop improvement. And this lab, it cuts across roots, tubers, bananas, millet, sorghum, legumes, so basically all legumes, other than peanut and soybean. So we have a lot of opportunity to scale up. Steve Krasovich is the director, who is my former postdoctoral mentor, and Jesse Poland is a co-PI along with me, and Jesse is the one who's all the conversations with Jesse is kind of how I developed this Go High training material. So yeah, we'll be scaling up. So I'm very excited about that.
Can I comment?
Well, let me just add, maybe Jeff, the question I have is the way you envision what is being done in the Sauergen project. Is that, do you expect each of the country components to continue at the country level, or do you envision the regional approach that the 4 different countries are working together now?
Okay. Yeah, okay, I understand your question now. So I'm not going to answer that because I think this is the time that it's really going to be up to the partners. So I'll let them answer.
Okay.
Can I comment on that?
Can I comment?
Yes.
Daniel, let me comment something, and later you can add your vision.
Okay, Cyril.
Okay. Currently, with the WAP project, we have collaboration with all the NARS. We have implemented a network of NARS countries: Senegal, Mali, Niger, Burkina Faso, and Togo. And without any funding in the network, in 2 years, without any funding, we share materials. In sorghum, for example, we share 28 inbred line. Each country have contributed in average 5 varieties. We share together and evaluate in several localities around the 4 countries. Daniel can add something about that. And currently with the Desira project, we are continuing. So to say that without any funding, we are extending material, we are working together. And when we have fund now, we can do better. So we test already the network without any funding, and it worked in 2 years, 2019 and this year. I don't know if Daniel can add something about the network we implemented in the DPAVAO network.
I'm not sure oh, there, Daniel, do you want to contribute?
Yeah, I want to contribute on that. I think that Sarajan is embedded in the overall regional dynamic. SERVAS is leading in West Africa as the real center of excellence, and there are a lot of initiatives going on actually in this network for several crops, including sorghum. As the Civil Jata said, the regional network for variety testing and sharing is something that is very real, that is working. People are sharing material every year. People are evaluating the material every year, and people are sharing also data of the evaluation. That is very impressive. And all the data is stored in the common server database, the BMS, and each breeder can have access to the overall data that have been produced during the last 2 years. So based on this network and based on also Sarah's commitment in trying to reinforce the regional dynamic on the region, we wrote a project that is funded right now by the European Union called AB. So AB is a project involving 3 countries in West Africa plus 1 in Mali. It's a 8 million euro funding for 5 crops, and this is also for strengthening the regional network have been built these last 5 years in West Africa, and breeders will continue materials to evaluate the materials, to have also on-farm evaluation in the fields, and there is a lot of different activities such as breeding profiles, definition, and link to the seed system, etc., etc. So I'm quite confident that the Sawagen network and the Sawagen product will really all these efforts that we are trying to put in West Africa and beyond the Sawagen funding. I think that the breeders will continue to collaborate, to share and data and to share materials. I'm sure that is a very nice example on how different projects are contributing to build something that is very big and that goes upon a single project.
Great, thank you. I think it's your point is really important, that it seems like the Sawagen project to get started has leveraged additional funds that you've been able to access. Farhani, follow-up question?
You know, I'm glad to hear from the regional representatives that this concept is going to continue even if the SME project phases out. I just wanted to hear their views, and I'm happy to hear that they are on the right track and they will continue it. I just wanted a confirmation from them. I'm glad to hear that.
I'd have to say I'm too, and I'm really excited about the enthusiasm that is demonstrated by Cyril and Danielle, and I think is probably echoed by the other partners as well. Other questions for the team? Do you have any hands raised or anything on the chat you want to submit?
Yeah, Peter Madeline has another question, asking us to underline the concrete milestones on the towards the development of sorghum varieties that are adapted, adopted, and appreciated. What specific outcomes do you expect to achieve in the next 4 years? What metrics will you apply to assess success? Thank you for bringing this back up to the discussion. Okay, so going back to this, I'm going to say that we will not be able to evaluate outcomes in these 4 years. Okay, so this adopted, appreciate, true outcomes out in seeds that have gone through the seed system, we're not going to be there in 4 years. So now outputs towards those goals, just as you asked, you know, those concrete milestones towards those outcomes is the output. What's going to be possible in the next 4 years? So going back to our AOP, we are now here. What's highlighted in yellow. We get part of year 5. I would say definitely in Senegal, we have a strong expectation that by the end of the project period, we will have got to the point of testing hypotheses on the appreciation and the adaptedness of those candidate varieties. Cyril is moving very quickly. He's working three generations a year, and he's using Ndiaga's Cisse's trick of starting on multiplications while the rest of the assessments are moving forward. The other teams that are you know, younger programs or programs that are still at the stage of building more capacity, this is maybe will be a stretch goal to get to that point where we're evaluating by the end, but we're currently on track, especially with the mini-NAM. So maybe with that, I will hand it over to anybody on the team who wants to say more about that. Cyril, do you want to say more about that?
Yes, what I believe is for the mini-NAMs, I evaluate them this year in 3 locations, and I'm analyzing the data. Now I identify in Senegal 10 promising inbred lines that combine yield and tolerance to grain mold. And this coming rainy season, I will carry out PVS, and maybe before the end of this project, I hope I will have something to register. Yeah.
And then Peter asked about metrics, and I'll just highlight that that's one of the reasons why the product profiles are so important. I'm just going to bring up at least the latest draft that I have on the product profiles, and I'll say these are not the final product profiles. We are breaking these down and testing many of the components of these. But the idea with the product profiles is that the metrics are literally there, you know, beforehand, a priori, and that each element of those metrics can be is a hypothesis about stakeholder demand that can be tested via participatory approaches, you know, surveys, PRAs. So, you know, not every single element that we're going to be able to test, but the metrics are baked in to the product profiles.
Peter, follow-up question?
No, I would just like to express appreciation for the very insightful and transparent response that Jeff gave. I want to underline again how impressed I am with the overall conceptual framework that you're working within and your approach in pulling together breeders from the 4 NARS. And I think recognizing the long-term nature of development of improved varieties, I think your response to Burhani's question is really key. How these platforms, this conceptual framework, this this culture of collaboration across national programs is so key to ensure that the momentum is retained and that you can achieve your outcomes, say in year 6, year 7, year 8. So I really appreciate your response, and again, I'm very impressed with your approach. Thanks.
Thanks, Peter. Thanks, Bettina, for the support, and I appreciate all the positive feedback on what to maintain, and I continue to welcome the critical feedback on what we should change. I pay close attention to all of it, and the team does as well.
From the management entity, Brahani, and everyone out there, I think that the Crop Improvement Lab has been invited to attend these meetings virtually. I think I've seen participation on previous presentations. I'm not sure if they're here now, outside of Tariq's, who you just mentioned, Jeff. But yeah, we're trying to build that continuity to ensure that what has been established in this program has opportunities to carry forward in other incarnations, whether they be from USAID or from the European Union funding that was described by Danielle. So more questions from the audience. Well, everyone, it doesn't seem like we have any hands going up or anything coming through the chat box, so we can pull this session to an end. Thanks very much.